Hi students, welcome to chapter 22, which is the lecture on blood vessels. Primarily what we're going to focus on today are the vessels and the walls of the vessels. What types of tissue are these body tubes made up of, and how do those tissues change based on the size of the vessel and based on the function of those different vessels. We'll learn a few other things as well. We'll learn about fetal circulation, and we'll learn a little bit more about the blood supply to the liver. That blood supply to the liver is referred to as the hepatic portal system. So that's where we're going today. I'll just remind you of a couple of things and bring up a couple of new things here. I don't know if I mentioned it in the last lecture, but the heart plus the blood vessels make up the cardiovascular system. And this is what we refer to as a closed system, meaning there isn't any place where blood is added and there isn't anywhere where blood is leaving the system, unless, of course, there's an injury. So you've got blood circulating around in this closed system of tubes and, of course, the heart. We learned in the last lecture that blood gets pumped from the heart into both the pulmonary and systemic trunks. Remember, the pulmonary trunks is the blood vessel that carries blood to the lungs, or the first blood vessel that's going to be carrying blood to the lungs. And the systemic trunk is also called the aortic trunk. That's the main vessel that's going to be carrying blood away from the heart to the head and neck and to the rest of the body. And remember, that's going on simultaneously. Remember that the heart beats asynchronously, where the atria contract and then the ventricles. Atria and then ventricles, that asynchronous beating. Two terms that you may or may not be familiar with based on your experience in a clinical setting are systole and diastole. So since we're talking about blood being pumped, and the only way that blood can get pumped or moved is through the contraction of the heart itself. And remember, the atria contract and the ventricles contract. So there's a way that there are terms that we use to refer to what's happening with those chambers. So systole is the period of cardiac contraction, or where a chamber, could be an atrium or could be a ventricle, is ejecting blood into another chamber or into an arterial trunk. So let's review this real quick. The atria, remember, are pumping blood into the ventricles. So the right atrium and the left atrium, when they contract, they squeeze or eject blood into the ventricles. Whereas when the ventricles contract, they are going to eject blood into an arterial trunk. So if you think about the right ventricle, for instance, the right ventricle, oops, I can't spell. Yes, I can. Goodness. So the right ventricle is going to pump blood into the pulmonary trunk, and the left ventricle ejects or pumps blood into the aortic trunk. The aortic trunk is the same, essentially, as the aorta. So that term, systole, is used to refer to when the chambers are contracting. But when one set of chambers is contracting, the other is relaxing. So, for instance, when the atria contract, the ventricles are relaxing. Or when the ventricles contract, the atria are relaxing. So there's another term that we refer to, or we use when we're talking about the chambers that are not contracting. When they're not contracting, they're filling back up with blood in order to prepare for systole again. So when a chamber is filling with blood, it is referred to as being in diastole. So you could say atrial diastole, and that would mean that the atria are filling up with blood, getting ready to contract again, or in other words, getting ready for systole. Okay, before we move on, I want to kind of explain the difference between an artery and a vein and make sure it's really clear that you understand why the book and most of the models will show arteries and veins in red and blue, and when there are exceptions. There are a couple of exceptions. So an artery is a blood vessel that's carrying oxygenated blood away from the heart. So if you have difficulty remembering which is which, you could think the A for artery means away. So arteries are carrying oxygenated blood away from the heart, normally. Not every single artery does that, and I'll tell you about the exception in a minute. And then veins are returning blood back to the heart, and usually that blood is lower in oxygen, or you could say deoxygenated. You could also say high in carbon dioxide. There are a couple of ways you can refer to that blood. So arteries carry blood away, veins carry blood back to the heart. Now there are some exceptions, and the exceptions exist in the coloration of the blood vessels in your textbook, in your models, and almost everywhere you look. The red color that is usually representing arteries isn't just indicating, or is not meant to indicate that it's an artery, though it usually does. The red coloration means that, or is referring to the fact that the blood is oxygenated. And most arteries are carrying oxygenated blood, with the exception of, let's move down here, the pulmonary arteries. Remember, the pulmonary trunk is carrying blood away from the right ventricle. That pulmonary trunk is going to split into the pulmonary arteries. That blood is being carried to the lungs, and the reason it's being carried to the lungs is because it's high in CO2. At the lungs, the blood, the CO2 in the blood will diffuse and it will be exchanged for oxygen. So oftentimes the pulmonary arteries, even though they're arteries, are going to be colored blue. In your textbook, in your models, they're going to be colored blue. That does not mean they're veins. It just refers to them, those vessels, carrying deoxygenated blood. And then the same is true for the veins that are carrying blood back to the heart from the lungs. So they're veins because they're returning blood to the heart, but they're going to be colored in red because these veins this blood has just exchanged CO2 for oxygen in the lungs. So pulmonary veins are carrying oxygenated blood back to the left atrium. So try to recognize that in your models and in your textbook pictures. Yes, you can say that usually arteries are colored red and usually veins are colored blue, with the exception of the pulmonary arteries and the pulmonary veins. So keep that in mind. Okay, as another reminder, blood vessels are going to be divided up into the vessels that are carrying blood to and from the lungs. Those are called pulmonary vessels. And the blood vessels that are carrying blood to and from the rest of the body would be referred to as being part of the systemic circuit. The names of vessels are actually pretty easy for you to remember or should be, because we covered a lot of these terms in the very first unit of the class. So usually vessels change their names based on where they're found, so that they're proper anatomical descriptions. So you might see this quite readily. For instance, the axillary artery or vein. You know that you would look for that in the axillary region, in the armpit. You'd look for the brachial artery or vein in the upper arm, and for instance, the radial or ulnar arteries or veins. You'd look in the forearm for that. Other examples, for example, the femoral artery, you would look for in the thigh. What about intercostal arteries or veins? You would find those between the ribs. So, although there are a lot of different arteries and veins that we ask you to identify, their names should be easier to remember because you already know these terms. Arteries and veins come in a variety of sizes that will have a variety of layers of tissues associated with them. But regardless of the size, arteries and veins that are carrying blood to and from the same region are going to lie right next to one another. So for instance, if this is an artery, you could choose any name you want. This could be the axillary artery, for instance. That's going to lie immediately adjacent to, oops, that doesn't look good, immediately adjacent to the brachial vein. And this serves a couple of purposes, and one of them is heat exchange. So if you think about arteries carrying warm core blood away from the heart, or away from the torso, here's the heart, and what happens to that blood as it reaches the periphery, it cools down a little bit. And it's losing heat. That blood is losing heat as it's traveling towards the periphery. And it's primarily losing heat to the environment, but a little bit of that heat is lost to the adjacent vein. And what's that adjacent vein doing? Well, it's carrying cooler blood back to the heart in the opposite direction. And so some of the heat that's lost from that artery gets exchanged, it doesn't get exchanged, but it gets taken up by the blood in the vein. And this is what we call countercurrent exchange, or countercurrent heat exchange. And so this is one method that we're able to regulate our body temperatures. So if you imagine, for instance, having been out in the cold for several hours, and you're really cold, and if you think about it, that blood, and I'm just going to make up a number, I don't know if I'm just going to make it obvious, an obvious example. Let's say you've been out in the cold for a really long time and your body temperature has fallen to 97 degrees. That's not possible. I think that you would be I don't think you'd be alive if your body temperature was 97. But let's just say, for example purposes, you've been outside for a really long time, your body temperature has dropped to 97 degrees. And that blood that's being carried in this vein coming back from the periphery is 97 degrees. The blood that's traveling in the artery, for instance, let me just change my pen color here. The blood that would be traveling in the artery would be, let's say, 98.6. And so that blood that's traveling in the artery is going to lose a little bit of heat to the blood in the vein. And this is going to gradually warm that blood up as it makes its way back to the heart and the core. So this prevents wild swings in temperature. It helps to maintain homeostasis in your body temperature. So with these blood vessels, most of the blood vessels are paired, meaning you have a left axillary artery and a right axillary artery, or a left femoral vein and a right femoral vein. And the exception is immediately near the heart. So for instance, you don't have a left aorta and a right aorta. There isn't a right pulmonary trunk or a left pulmonary trunk, or a right superior vena cava and a left superior vena cava. Immediately at the heart, those vessels are not paired. So as I've mentioned in the last two systems we've covered, digestive and respiratory system, the body tubes that belong to each system for the remainder of the course are going to have layers associated with them. And remember that when we're designing, let me rephrase that. When we look at body tubes, we always find that the tube itself has an epithelium lining the lumen. When we're describing body tubes, or their layers of tissue, now something has changed a little bit. This simple squamous that's lining this vessel right here, that layer, we say that that's the superficial layer because it's facing the lumen. So with body tubes, the layer of tissue that lines the lumen is referred to as superficial. And then as we move outwards, the tissues are referred to as being deeper. So it's backwards from what you have learned in the past. Anyway, the layers of tissue that are found in blood vessels share a similar structure-function relationship to the tubes that you've seen before. All hollow body tubes, all hollow body organs are going to have some type of epithelial tissue lining the lumen, and then the successive tissues that we find from superficial to deep or superficial outwards will share also a very similar pattern. There's going to be some connective tissue, there's going to be some muscle tissue, there's probably going to be some more connective tissue, or an adventitia. And so the same is true here when we're looking at blood vessels. And the layers are called tunics, or tunica. And even though this picture right here is showing you a vein, these three layers are present in arteries as well. So let's have a look at those different layers, and we're going to go from deep to superficial. So we're going to move from outward towards the lumen. And the outermost layer in both arteries and veins is called the Tunica Externa. And that's mostly made of what we call fibrous connective tissue, or you could just say dense connective tissue. It's kind of a strange one in that it's not quite areolar connective tissue, but it's not quite dense irregular. So we just use this blanket term and we call it fibrous connective tissue. The role of that layer is to attach blood vessels to the deep fascia. So we could be anchoring blood vessels to nearby connective tissue, or we could, let's say, what can I use as an example? We'll use a nerve. Let's say there's a nerve passing right next to that blood vessel. And of course, that nerve would be solid, right? That's not a hollow body tube. But what surrounds a nerve? What's that outer layer called in a nerve? Can you think of it? And this just totally does not look like a body tube anymore. Or not a body tube, but a nerve. So pardon me. Remember, the outermost layer of a nerve is called Epineurium. And that's made of a fibrous connective tissue as well, or a dense irregular connective tissue. So these two layers on the outside of the vein and on the outside of the nerve are going to anchor those two structures together. Or there could be a muscle that this vessel is passing through, and the tunica externa will weave into the epimysium of the muscle. So this tunica externa really just allows blood vessels to attach to things surrounding them. And then next up is the tunica media. Let me erase some of my previous drawings here. The tunica media is the middle or intermediate layer, and that intermediate layer, that middle layer, is going to be made of smooth muscle right here. So that red line that you see, or the red donuts encircling the vein, that's smooth muscle. And what that smooth muscle is going to do is allow the vessel to decrease its diameter or increase its diameter. So when the vessel increases its diameter, we say the blood vessel's dilated. We often use the term vasodilate. Vaso means vessel, so vasodilation means blood vessels get wider when that smooth muscle relaxes. Or when that smooth muscle contracts, it constricts, or the diameter of the blood vessel narrows. We call this vasoconstriction. You'll learn more about this in physiology. But this smooth muscle is going to allow the blood vessels to regulate blood flow to any region of the body. And this helps with things like temperature regulation and or gas exchange or regulation of diffusion in capillary beds. We'll talk more about that later. Because it's smooth muscle, it's regulated by the sympathetic portion of the autonomic nervous system. So this is not a muscle that you have control over. It's going to be contracting or relaxing based on the autonomic nervous system. Next up then is the most superficial lining, and that's going to be here. And this is going to have two sublayers to it. So this layer itself is called Tunica Interna. Your book may call it endothelium. Don't call it that, and I'll tell you why in a second. But I just put that there so you know that sometimes those two terms are used interchangeably. The Tunica Interna is made of a layer of simple squamous, and that's what you see there in purple. But deep to that, let's have a closer look. Deep to that is that yellow layer that you see right there. So right here, and that's a layer of areolar connective tissue. So the Tunica Interna has two layers, simple squamous plus an areolar connective tissue layer. And you could probably guess what that areolar connective tissue layer is doing. It's binding the epithelium to the underlying Tunica Media, which is right here. Okay, so that simple squamous that you find there is referred to as endothelium. Kind of sounds like epithelium, but we call it endo because it's deep inside the body. Sometimes authors will use the term Tunica Interna and endothelium interchangeably, but really endothelium only refers to the simple squamous and does not include the areolar connective tissue layer. So remember that let me change pens here. There are two sublayers. One is the simple squamous layer. It's simple squamous epithelium, but that's what we refer to as endothelium. And then the second layer is a layer of areolar connective tissue. Okay, so two sublayers making up the Tunica Interna. Saying Tunica Interna accounts for both the simple squamous and the areolar connective tissue. If you just say endothelium, it only refers to that simple squamous. So just so you know. The function of having the simple squamous here in lining the lumen is that it reduces friction from the blood passing through the vessels. So if you can imagine blood passing through these vessels right here, the last thing we want to do is have blood products or blood cells or anything that's being carried in the blood get caught up on the endothelium, because that can snowball very quickly and reduce blood flow, which can be a problem. So it can also slow down blood flow, and what will happen when blood slows down is clots begin to form. Blood starts to coagulate. So we want to make sure that there's a nice slippery surface for the blood products to pass over. So now that we've gone over the different layers that you can find in both an artery and a vein, if we look at the histology of blood vessels, you can see those layers pretty easily. And these different types of tissues that you see here should be very much recognizable to you. So over on the left, let's just go ahead and look at those layers. So the Tunica Interna, sometimes, don't kill the messenger, sometimes Tunica Interna is called Tunica Intima. Just depends on the authors. So these two layers, the layer of areolar connective tissue and the layer of simple squamous, are going to make up the Tunica Interna, or another way to refer to that is Tunica Intima. And then you see this really thick layer of smooth muscle, and it's only this thick in an artery. And the reason for that is arteries are under a great amount of pressure that has been generated by the heart. So arteries need to be able to withstand the pressure that the heart generates and also expand as blood passes through. The reason that the heart generates so much pressure is that heart needs to be able to pump blood against gravity through the neck and up to the head, and also pump blood all the way to the tips of your fingers and all the way to the tips of your toes. So that those vessels need to be able to accommodate the pressure that the heart generates. So you have a much thicker tunica media in an artery than you would in a vein. And this is one of the reasons why arteries, even when there isn't any blood inside, they maintain this nice round shape. And that's very different than when you look at a vein. A vein, at least in this underneath the microscope right here, is kind of more of a pancake shape. It's kind of collapsed on itself, especially when doesn't have blood inside. And look at how much thinner the tunica media here is in that vein. Or if you look all the way over to the right, it's much thinner. And so veins do not carry pressurized blood like the arteries do. They're not under the same amount of pressure. Veins, as you recall, are carrying blood back to the heart. And there's no pump at your feet. There's no pump at your fingertips. So this blood that's carried in veins, or the veins, aren't under much pressure, so they don't require a nice thick layer of the tunica media. And then the last layer, let's go back to the picture on the left. The last layer is going to be that tunica externa. And that's going to be fibrous connective tissue. Sometimes that tunica externa is also called adventitia. You can use either one, I don't mind. But those are the layers. One of the tricks to determining an artery from a vein underneath the microscope is, of course, looking at the nice circular shape of the artery, looking at the thickness of the tunica media, but If you were to look closely or increase the magnification and specifically focus on the Tunica Interna, you'll see that the Tunica Interna in an artery is kind of got this scalloped edging right here, whereas the Tunica Interna in a vein is totally straight. So that's another way you can tell the difference between the two, or confirm a difference. So we've learned about the different layers that make up blood vessels and what type of tissue those layers are made up of. And we've also learned or looked at the histology of both arteries and veins. And we're able to determine some of the differences or see some of the differences. Now what I'd like to go over with you are the different sizes of arteries and the different sizes of veins. And I want you to just understand the kind of overall purpose of having blood vessels. The heart is pumping blood, and that blood needs to get to all the trillions of your cells. And what's being carried in that blood are things that each one of those cells need to live. Oxygen, glucose, enzymes, hormones, a number of different types of amino acids and or polypeptides or proteins, different types of carbohydrates. You name it, the heart is pumping that blood filled with all of those things that are going to be required by cells. But the blood vessels, many of them, are simply conducting structures, just like we saw in the respiratory system. They're tubes that are conducting blood to some area, and then when blood finally reaches its destination, that's where exchange gets carried out. Just like with the respiratory system. Once you finally got down to the alveoli and the respiratory bronchioles, that's where gas exchange was occurring. And the reason why gas exchange was able to occur is because the walls of the tube were thin enough to allow for it. And the same is true with blood vessels. We'll find that the majority of blood vessels, I shouldn't say the majority, the larger vessels, look at their walls. Look at how thick this wall is right here. No exchange is going to occur through that wall. No exchange is going to occur through this wall. Very little exchange is going to occur through this wall. It's not until we get down to the capillaries that are just made of simple squamous that exchange is going to occur. Not just gas exchange, but exchange of water, ions, nutrients, you name it. So what we see is that blood vessels are going to vary in the layers that they have on them. So for instance, an elastic artery is going to be one of the major arteries that's carrying blood away from the heart. So they're showing you in this example right here, the descending aorta, that's going to have all three layers. That's an elastic artery. That elastic artery is going to be able to withstand the pressure that the heart is generated. You see it's got a thick Tunica Media right here. It's got a thick Tunica Externa to anchor it to nearby structures in the body. This is clearly not an exchange surface. It's way too thick. But as that elastic artery continues down the body, for instance, these blood vessels, all blood vessels, gradually as they go towards their destination, are branching and getting smaller and becoming more numerous, and branching and becoming smaller and becoming more numerous. And as that happens, as those blood vessels get smaller and smaller and they reach their destination, you'll see that the layers change. So you don't see a huge change here in the muscular artery, but this is a vessel that is smaller than an elastic artery. these muscular arteries are carrying blood out to the arms and the legs, or out to skeletal muscle. And you see that right off the bat that the muscular artery is a little smaller than the elastic artery, but the muscular layer right here, that tunica media, is thicker than it is in an elastic artery. So the vessel itself is smaller, but tunica media is thicker. Those muscular arteries will branch and get smaller and become more numerous, and they become structures called arterioles. And if you look at the arteriole now, the tunica externa, the outermost layer, is completely missing. And not only that, the tunica media, the muscular layer, is just going to have little strips. Let's look at this, little strips of smooth muscle left over, right? So we've gotten rid of the outermost layer, that tunica externa. You see, we still have the endothelium, you still have that tunica interna. So even though the vessel is smaller and has fewer layers at this point, the vessel still is not an adequate exchange surface. That doesn't occur until you shed that muscular layer, that tunica media, completely, and when that happens, it becomes what we call a capillary. Capillaries just have those endothelial cells, and those endothelial cells are squamous in shapes. We've got a simple squamous epithelium that's going to allow for gas exchange. I'll talk a little bit more about that in a second. And you've got capillaries here, you've got capillaries here. So the capillaries kind of come together in a network. This is kind of like the U-turn right here, right? That structure is called a capillary bed. And those capillaries now that begin the return route of blood back to the heart need to add layers of the blood vessels or layers of tissue back on. So now you've got many small capillaries merging together to become slightly larger capillaries. So you'd have capillaries coming in, and they all merge together to create this larger vessel. And then, as you can see, more vessels merge together, and so you finally make some of the larger veins. And so as the blood returns through these vessels back to the heart, tissue layers are added on. So in the veins, what we see is once there's a tunica externa added back on, that's what we a venule. So it's a little bit larger than a capillary, and they're going to be less numerous and have tunica externa on them. Many of those venules will merge together to become what we call a medium-sized vein. And those medium-sized veins, as you can pretty clearly see, now have a tunica media, although the tunica media is much thinner than it would be in an artery. So you've essentially got all three layers now. You've got all three layers added back onto the vein. So clearly, again, not gas exchange structures. And then many medium-sized veins are going to merge together to create what we call large veins. And large veins now are going to have still all three layers, and that tunica externa is going to even be a little bit thicker than it was in the medium-sized vein. These large veins and medium-sized veins have specialized structures in them called one-way valves or venous one-way valves. That's something that the venules do not have, and I'll tell you what those venous one-way valves do here in just a second. So on this slide is written essentially everything that I just mentioned on the previous slide, so I won't reread this to you. The only thing I didn't cover on the previous slide is that with the capillaries, so let's look down at the bottom here, with the capillaries, there are different types. So there are what we call continuous capillaries. Continuous capillaries their wall, as you might expect, is continuous. The endothelium is continuous, and products are still able to leak out between the squamous cells and through the squamous cells, but most of the capillaries in the body are continuous capillaries. But there are exceptions in certain organs, so fenestrated capillaries, for instance. Fenestrated capillaries are very porous. They're missing sections of the endothelium. So don't laugh, but I'll just draw it in red. Let's say, for instance, here is a capillary. And rather than having a continuous tunica media, you'll find that it's got big holes in it. And those holes allow for products to diffuse out of the capillary. And then another type of capillary is what we call a sinusoid. Remember when we were learning about the liver and the digestive system, that it has those sinusoids that pass through the lobules of the liver? So a sinusoid, let me see if I can draw this. Let's say here's the blood vessel. A sinusoid is going to have large pieces of the vessel missing completely. And what this is going to allow for is more blood, or not more blood, just blood to physically leak out of the vessel. Remember in the liver how the blood leaked out of the vessel and saturated or kind of immersed the hepatocytes in blood so the hepatocytes could do their job to modify that blood? Really, the only place that you find sinusoids are in the liver. Fenestrated capillaries you can find in a couple of places, like the choroid plexus or in the kidneys that we'll learn about next time. So there are variations on the capillaries, but I don't want you to think that a continuous capillary becomes a fenestrated capillary and that a fenestrated capillary becomes a sinusoid. No, that's not the case. Fenestrated capillaries and sinusoids are just different. Okay? So we can see the difference between a continuous capillary and a fenestrated capillary, where over on the left is that continuous capillary and you don't see any holes within that squamous cell, right? So the wall is continuous. Now remember, products are still going to be able to leak out, like oxygen. Products are still going to be able to leak in, like CO2, right? But if we look over on the right, in a fenestrated capillary, more products are going to more easily leak out through these fenestrations, these little black dots that you see here. All of those are teeny tiny little holes that are going to allow for greater amounts of material to either pass out of the vessel or into the vessel. So we kind of just left off here with understanding the arteries. And what I want to point out, and I guess we went over these small venules and how they come together to make large veins as well. But what we haven't covered yet is what happens in between. What's this area here? That area there is what we call a capillary bed. And it's at capillary beds where exchange occurs, where diffusion occurs. So let me erase all of this. So remember, at the beginning of the lecture, I mentioned that most of these tubes that belong to the cardiovascular system, they are conducting structures. They're conducting blood from one place to the next. But there is a destination for that blood. And the destination for, not necessarily the blood, but the products that are being carried in the blood is at individual cells, right? So if we look at this capillary bed, it's kind of this network of vessels right here. Let's say, for instance, we'll make our cells yellow. Here's a body cell, and imagine body cells being packed all around here, right? So it's those structures that require nutrients and oxygen. They also require waste products like CO2 to be removed from the area around them. So if we look at capillary beds are where gas exchange occurs. So if we're thinking about blood flow, for instance, blood is going to pass down from this or through this arteriole and ultimately make its way into a capillary bed. And that blood's going to pass through various locations in the capillary bed. There are multiple ways in which blood can pass through. We'll just follow it through here. And you can see here now that these vessels are continuous capillaries. Look, their walls are really thin. And what's going to happen is, for instance, oxygen is going to diffuse out, and it's going to make its way towards the cells that require it. What's also going to happen, though, is those cells are creating CO2 as a byproduct of cellular respiration. So CO2 is going to diffuse into the blood vessels. And it's not just gas exchange, right? There's going to be glucose and ions like sodium and potassium and magnesium. All of those things are going to diffuse out of blood vessels to the area that we find the cells in. And cells are going to be secreting waste products, and those waste products are going to want to get back into the blood. That blood then, as it continues on, is going to, from all of these areas, eventually merge together into venules. And then many venules are going to merge together to become a vein, and so on. So essentially what a capillary bed is it's an area where exchange occurs, but it's also a U-turn for blood. Blood now is going to start making its way back to the heart. So via arteries, blood is leaving the heart. It's at the capillary bed where the gas exchange occurs. That's also the U-turn. And then blood then starts to collect in the venous system and make its way back to the heart. What's interesting about these capillary beds is they have some structures. Let me see, I'm running out of colors to use here. I guess I'll just use red. These capillary beds have what we call pre-capillary sphincters, these structures. And they're essentially circular muscles. That's what a sphincter is, right? Essentially circular muscles that are going to contract and relax to regulate blood flow through the capillary bed. So you don't have blood just freely all the time passing through, I'll use green, passing through and then returning. That blood flow is totally regulated by the pre-capillary sphincters based on what the needs of the surrounding tissue is. So for instance, if this tissue out here has plenty of oxygen in it, what may happen is the pre-capillary sphincters might contract and close, only temporarily, of course, because this area has got enough oxygen. And after some time, then, what will happen is the pre-capillary sphincters will open up and blood will make its way through, and just for instance, oxygen diffuses out again. So these pre-capillary sphincters are constantly contracting and relaxing to regulate blood flow through this capillary bed based on what the tissue's needs are. So this slide here, I've already mentioned that venules come together to make medium-sized veins, and medium-sized veins come together to make large veins. So this is just a reiteration of what I went over in the picture a few slides ago. So I won't reread this to you. So now we've learned about the layers that we find in arteries and veins, and that at capillary beds is where exchange occurs. Now, earlier I said that veins aren't under as much pressure as arteries are. And that's because there's no pump at the feet or the tips of the fingers. So there are several ways in which blood moves back to the heart, especially if you consider when you're standing up or even sitting in a chair that blood has to be moved against gravity. So the body has several different methods that allow for blood to move back to the heart in the absence of a pump pushing it back to the heart. So the first thing I'd like to explain to you, or this first method, is what we call a venous one-way valve. And just temporarily, I'm going to move to the next slide. Go back to this slide. So we've covered the valves. We know what they look like. We know what they do. And also we know that skeletal muscle, not just the gastrocnemius, but skeletal muscle all over the body, plays a role in compressing the veins and moving blood through. Another method in which blood is moved through those veins and back to the heart is through the constriction and relaxation of that muscular layer, Tunica Media. The other two ways are a little bit different in that they are a little less obvious. One is the respiratory pump. So the respiratory pump, it works like this. Imagine taking a breath in, take a deep breath in right now. And as your lungs fill with air and your diaphragm depresses or puts pressure on the organs of the abdominal cavity, that's like squeezing the organs. That pressure squeezes the organs, kind of like squeezing a sponge, and squeezing the blood out of those organs. And that acts as a method to move blood through organs and through veins. The other method is what we call cardiac suction. And remember that the cardiovascular system is a closed system. And so whenever blood leaves a chamber or vessel, a small vacuum is created. So consider, for instance, when the right atrium contracts. When it contracts, it ejects its blood into the right ventricle. But as it relaxes, it's going to pull blood in from both the superior and inferior vena cava. So again, it's kind of like squeezing a sponge. When the atrium contracts, that's like squeezing a sponge underwater, and you're getting all the water out of that sponge. And then when the atrium relaxes, it's like your hand releasing the sponge underwater, and that water gets pulled into the sponge. And so every time a chamber is relaxing and filling with blood, it's pulling blood into the heart because the cardiovascular system is a closed system. And show you what a venous one-way valve looks like. A one-way valve is an extension of the endothelium into the lumen of the vessel, and it literally creates a valve. A valve or blood can only move in one direction, and blood can only move through the valve back to the heart in this direction. And once that blood moves through the valve, the flaps of the valve here, they close back up. And so that prevents blood from flowing back to where it came from. So you'll find these one-way valves in large veins and medium-sized veins. But in order for that blood to move and these one-way valves to do their job, the veins have to be compressed. The blood within those veins has to be squeezed upwards. So if we look at the picture on the left, this is a vein that's passing through the gastrocnemius. And over on the right, the authors are showing you when that gastrocnemius contracts, it compresses the blood vessel. So this contraction of skeletal muscle assists the blood in returning back to the heart by squeezing or compressing those veins and pushing blood back up towards the heart. And then what prevents blood from flowing backwards are those one-way valves. We'll move on then to a couple of other topics and then we're almost finished. Okay, students, we're going to switch gears here a little bit. When we were learning about the digestive system and how food and nutrients are absorbed into the bloodstream, you learned about the hepatic portal vein, and that the hepatic portal vein is draining blood from the GI tract and carrying it directly to the liver. Well, there are other blood vessels that contribute to create what we call the hepatic portal system. And so what we'll do is we'll go over those blood vessels that help to create that. And we'll also look at a picture of those vessels too, so you can kind of get a better idea. Let me first start off with letting you know that there are blood vessels here listed for you that are responsible for blood supply to the liver. And the hepatic arteries, these are arteries that deliver oxygenated blood to the liver. The hepatic portal vein and the hepatic veins, they are not carrying oxygenated blood. It's deoxygenated. What those two veins are carrying are blood that either needs to be modified by the liver or blood that has already been modified by the liver. So they're not delivering oxygenated blood. So the hepatocytes need a source of oxygenated blood. The hepatic portal vein is carrying nutrient-rich blood into the liver to be modified. And the hepatic veins are draining that blood that got modified in the liver. They're going to drain that, I'll just call it modified blood, from the liver and into what we call, and you should know, oops, not the superior, shame on me, the inferior vena cava. So the hepatic portal vein, you guys, doesn't just magically arise as this one vessel that collects from every area of the GI tract. It's actually made of many vessels coming together. Earlier in the lecture, we learned about how venules come together to make medium-sized veins, and medium-sized veins come together to make large veins. The hepatic portal vein is a large vein, and there are many medium, small, and small veins that contribute to the creation of the hepatic portal vein. So before I move on to the blood vessels listed below here, let's have a look. Okay, so let me point out to you first the the hepatic portal vein. And that hepatic portal vein is this structure here. But you see it has a number of tributaries that are draining into it. So for instance, the right gastric vein is draining into the hepatic portal vein. There is the splenic vein right here. That's going to drain its blood into the hepatic portal vein. There's the inferior mesenteric vein. That inferior mesenteric vein is draining blood from the descending colon. So that also comes together to make the hepatic portal vein. And then there's the superior mesenteric vein that drains the ascending colon and the majority of the small intestines. That's here. So you can see how all of those veins come together. Oh, one more, I forgot. The left gastric vein is going to drain into the hepatic portal vein. And then you can see the hepatic portal vein continuing on, and then it branches several thousand times to become those little branches of the hepatic portal vein that you learned about when we were studying the liver. So all of those vessels, those veins that ultimately drain into the hepatic portal vein, are part of what we call the hepatic portal system. Now in the background, you can just barely see these veins right here. Those are hepatic veins. So what these veins are doing is they're taking blood that's been modified from the liver and dumping it into the inferior vena cava. So if you think about it, I'm just going to use red, but that doesn't mean anything with regards to oxygenated or deoxygenated. It's just going to be an easier color to see. If you think about all the nutrients that are being absorbed from the small intestine, water that's being absorbed from, excuse me, the colon, anything that's been absorbed by the stomach, if we want to track blood flow, and I'll just do it from one place here, blood that's carrying nutrients is going to pass into that hepatic portal vein and then be distributed over the hepatocytes, right? And then the hepatocytes are going to modify that blood, and ultimately what happens is the hepatic veins are going to drain that newly modified blood. They're going to drain that newly modified blood. That newly modified blood then ends up in the inferior vena cava, and that inferior vena cava will take that blood to the heart. So those vessels that make up the hepatic portal system are really important for transporting nutrients to the liver. The liver modifies the blood and regulates nutrients, and then sends that blood into general circulation. And then lastly, I'd like to cover some differences between fetal circulation, blood flow in the fetus prior to birth, versus what we see in class in the adult. During fetal development, the fetus is anchored to mom via this structure right here. And that structure is called the placenta. That's actually anchored to the inner lining of mom's uterus. The inner lining of mom's uterus is very well vascularized, and so is the placenta. So all of these vessels that you see, that's a bad color, let me switch. All of the vessels that you see here coming from the placenta are going towards the fetus. So they're carrying blood towards the heart of the fetus. And then everything, all the vessels that you see here in blue are carrying blood back to the placenta. So this is the fetus's source of nutrients and oxygen from mom. Even though the fetus has its own blood vessels, it's still carrying out exchange of nutrients and gases through mom's blood. And if we look at some modifications here, you'll see, hold on, let me erase some of this stuff. I'll be right back. Okay, so because the fetus is carrying out gas exchange via mom and nutrient exchange via mom, we're going to see some modifications to the liver and the heart. So first of all, let's look at modifications in the liver. The fetal liver, at least not up until very near birth, the fetal liver is not functional. And the blood that is circulating through the fetus has been modified by mom's blood. So mom's liver is doing the work of the fetal liver. So there's no reason for blood that's coming in from the placenta to travel into the fetal liver and be modified. The fetal liver isn't functional, and that blood's already been modified anyway. So that blood's going to pass directly behind the liver, the fetal liver, and enter the inferior vena cava. And it does that through a structure in the fetus that we call the ductus venosus. And so that's a structure that once the fetus is born gradually shrinks and becomes much reduced until all that's left is a bunch of Tunica Externa. So it essentially becomes in the connective tissue separating the two lobes of the liver. One of those pieces of connective tissue inferiorly. You learned in the GI tract, or in the digestive system rather, is called the round ligament. That's literally what's left of one of the umbilical vessels from fetal development. Let's look at modifications in the heart. So we're going to look over here to the right because the heart is bigger over here. A couple of things. So the fetal heart is beating right around 6 weeks of development, but that doesn't mean that it's fully functional. It's still pushing blood, but remember the heart is really meant to push blood to or through the systemic circuit so all the cells of the individual can be nourished. And that's still true here. But what the fetal heart cannot do is push blood through the pulmonary circuit to the lungs because the lungs are filled with fluid. So there's no way that inside of the uterus the fetus is able to carry out gas exchange. The fetus is carrying out gas exchange through blood vessels and ultimately through mom's breathing. So let's just consider here blood coming into the fetal right atrium, making its way down to the fetal right ventricle. This blood has already been oxygenated by mom. And so the blood continues through in the normal pathway out that pulmonary trunk, but the lungs are filled with fluid. So that blood cannot go to the lungs, and there's no reason to because it's already been modified and gas exchange has already occurred. So there's kind of a block here where blood doesn't get pumped to the lungs. What actually happens is blood makes its way through a structure here that allows for blood to go immediately to the aorta and then through the systemic circuit. In the fetus, that passageway is called the Ductus Arteriosus. The picture that you're looking at over on the right is after delivery. After delivery, that Ductus Arteriosus shrivels up to become a strand of Tunica Externa. And in the adult, we don't call it the Ductus Arteriosus. In the adult, we call it Ligamentum arteriosum. And so that's going to be what's left over of that Ductus Arteriosus. One other modification that we find in the fetal heart is a passageway between the right atrium and the left atrium. So let me erase some of this so it's clear for you all to see. So again, I'm drawing on the infant's heart just after delivery, not over on the left as it is in the fetus, but only because this picture on the right is bigger and it's easier to see. So here's the right atrium, and here's the left atrium. And what you'll see is a structure here. It's a passageway. You don't see the opening on the other side, but imagine that there is an opening on the other side. That structure, when it's open in the fetus, is called Foramen Ovale. And Foramen Ovale allows for fetal blood that's already got oxygen in it, that's come from, it's been modified by mom. That fetal blood skips the right ventricle, and so it goes right from the right atrium over to the left atrium and then down into the left ventricle. Well, why does it do that? Well, some of that blood does go down into the right ventricle and then out the pulmonary trunk, but remember, that blood can't go to the lungs anyways, so why send it all through there? So some of that blood that's already been oxygenated takes a shortcut right to the left atrium. When an infant is first born, what happens is that first breath the infant takes helps to close the foramen ovale. It's actually not a wide open hole. At birth, it's a little, or during fetal development, it's a little flap. And then at birth, that flap should begin to seal up and prevent flow of blood between the two atria. But that doesn't always happen properly. And so you might know somebody, or maybe you were one of those babies that were born with a hole in your heart. Well, all of us are born with a hole in our heart. And for most of us, that hole seals up. But for some of us, it doesn't. And it used to require surgery. Now there's an oral medication that can be given to help seal that hole up. So eventually, in the adult, there's still an impression left between the atria. And that impression is called, not a hole, now it's just a groove, an impression, fossa ovale. So that brings us to the end of the Chapter 22 Vessels lecture. Thanks for tuning in. See you soon.